My name is Rolly Wang from Royal Institute of Technology, and together with my co-PI Yuan Li, we are presenting our digital pair project aiming to use digital tools to facilitate personalized medicine in rehabilitation. Impaired motor function is one of the major disabilities. Persons may lose their ability to perform daily activities and largely influence health-related quality of life. Management of a complex disability currently largely rely on rehabilitation. However, in clinical practice, the supervision and the evaluation of a rehabilitation motion pattern remain a medical and engineering challenge due to the lack of biofeedback information about the effect of the rehabilitation motion on the individual human biological tissues and structures. Therefore, the project aims to develop the digital twins, DT, of the personalized human neuromuscular skeletal framework, will allow carrying out a high-fidelity real-time analysis of non-measurable internal biomechanics quantities in the body tissues and bone structure in a non-invasive manner. As a proof of concept, we proposed a clinical-friendly digital twins framework that provides a real-time biofeedback to facilitate in neurorehabilitation training and intervention. However, to translate the power of the computational neuromuscular model from a research lab to clinical conditions, we face several major challenges. First of all, the model requires high level of personalization in respect to anatomical, physiological, and neurological characteristics to match the individual. Second, most skin contact sensors suffer from limited signal stability and discomfort during long recording. And last but not least, data input still largely rely on a lab environment, for instance, a marker-based motion camera system. We target these challenges in three perspectives. First, we will develop ultrasonic electrodes, which can record muscle electrical signals and ultrasound image simultaneously. This step can largely simplify the calibration procedure. Yuan will talk about our innovative sensor idea a bit later. The framework will incorporate data-driven and physics-informed approach, which can improve the reliability and the fidelity in parameter estimation. Also, we will use purely variable sensors in the framework, which will help to achieve the goal to be feasible applying in a clinical environment.
To get ultrasound transparent electrode, our strategy is to combine renewable resources-based ultrasound transparent substrate with biocompatible ultrasound transparent electrode material, that is PEDOT-PSS. Using scalable 3D printing technology, PEDOT-PSS could be printed on the substrate and result in ultrasound transparent electrode, that is the ultrasonic electrode here. The ultrasound transparent substrate we use are wood-based nanocellulose papers. Here shows the hierarchical structure of wood. Cellulose is one of the main components of wood, with stress in the order of 1 gigapascal. To make a nanocellulose paper, we first extracted nanocellulose from wood, followed by filtration and drying. The paper is further used for as a substrate for 3D printing, where the ink is PEDOT-PSS. Electrode pattern could be designed according to our demand. After 3D printing and drying, ultrasonic electrodes are obtained, shows here. Here are our preliminary results. We prepared two types of nanocelluloses from wood. One type of nanocellulose is temple oxidized SNF, which has a higher surface charge with better water-holding property, beneficial for long-time ultrasound transparency. To obtain this type of SNF, wood is under pulping, temper oxidation, and microfluidization treatment. Here is the atomic force microscopic image showing the SNF morphology. The diameters of the SNF are less than 5 nanometers. The other type of nanocellulose is called hollow SNF, which has better printability for the paper for further 3D printing, beneficial for stable electrical signal collection. For hollow SNF, we use prussic acid treatment followed by blending using kitchen blender. Similar morphology is obtained with less uniform nanocellulose diameters. Both types of nanocelluloses could result in strong transparent substrates with stress over 200 megapascal. These substrates are suitable for 3D printing of P.PSS. Here shows the 3 pairs of electrodes printed on hollow SNF paper.
We have also tested the muscle electrical signals and ultrasound image qualities using our developed ultrasonic electrodes while acquiring data simultaneously. Here you can see a comparison with the commercial electrodes when ultrasound image are acquired separately. Both EMG signals and ultrasound image look quite promising. In parallel, we are developing a real-time neuromuscular skeletal framework, which can be driven by different variable sensors. Our implementation of OpenSim RT can receive different type of sensor input, either visual alpha markers, a type of augmented reality-based marker, or IMU input, a type of electronic device using a combination of accelerometers, gyroscopes, and magnetometers, and use that to drive an inverse kinematics model to compute drawing angles during motion in real time. In the demonstration video, we fix the sensor input to the trunk of the skeletal model. As long as we have access to full set of IMUs, we can drive all degree of freedoms in the model in real time. Our ROS communications and infrastructure also allow us to perform real-time static optimization for estimation of individual muscle activations. In the animation, we can see the active muscle is illustrated in the red color.
This project is a multidisciplinary collaboration between the fields of biomass-based advanced materials design and the computational biomechanics of human movement. Our vision is to build a state-of-the-art feasible and reliable digital tool in personalized medicine in rehabilitation towards healthy life. Thanks for your listening. Please ask if you have any questions.